Finally, very different looking animals are actually built using similar genes and pathways. One of the most striking examples of this was originally found in research in Drosophila, the fruit fly. Drosophila has a whole set of key developmental control genes called Hox genes. These genes occur in clusters along the chromosome, and the expression pattern of the genes in the embryos is related to their position along the cluster. So the blue genes here at the one end of the cluster are expressed at the head end of the embryo that's shown there in the bottom right. The colored genes in the middle are expressed in the middle of the embryo, and the green genes at the end of the cluster are expressed in the tail region of the embryo. So those genes aren't just expressed at different anterior, posterior, head-to-tail positions. They're also required for the formation of the particular tissues that you find from the head to the tail of the adult Drosophila. So if you make mutations in those genes, you change the development of the characteristic tissues that should form along the anterior-posterior body axis. Well, surprisingly, although animals look very different, mice look very different from fruit flies, their anterior-posterior body axis is built from the very same sets of genes. So mice and humans, other vertebrates, have sequences that are closely related to these Hox genes of flies. The Hox genes in mammals also occur in clusters. The clusters are also expressed in patterns that are related to anterior-posterior development. Blue genes at one end of the cluster, they've actually duplicated, so there's more copies in mammals. But blue genes at the end of the clusters are expressed in the head regions of the embryo, shown at the lower left. The middle genes are expressed in the middle part of the embryo. The green genes at the end of the cluster are expressed in the tail region of the embryo. And just as in fruit flies, if you make mutations in these key Hox genes, those mutations alter the formation of the characteristic tissues that would normally form at each position along the anterior-posterior body axis of the mouse. So it looks like the head-to-tail axis of animals, even that look completely different, are in fact built by an ancient toolkit of these key developmental regulators called Hox genes. Similar toolkit genes have been discovered that control a range of other structures in diverse organisms. So the Hox genes for the anterior-posterior axis, other sets of genes for the dorsal-ventral axis, other toolkit genes for the left-right axis, and other toolkit genes for the formation of particular body tissues. To give an example of one particular tissue, eye development. So eyes, very interesting structures found in lots of different animals. They look quite different in different animals. So that's a human eye and a Drosophila eye. Human eyes has single light-gathering organ and lens. In contrast, insects have these compound eyes, thousands of little light-gathering organs all in a little crystalline array. The histology of the eyes is quite different. Single light-gathering lens for the mammalian eye and this array of independent little light-gathering units in the compound eye of insects. Although those eyes look very different, it turns out that the same toolkit gene is required for eye development in many different organisms. So this gene is called PAX6. If you make a mutation in one chromosome, one of the two copies of PAX6 during human development, you begin to lose particular parts of the eye, in particular the colored iris muscle that normally surrounds the pupil. So you can see there the eye of a patient with defect in one copy of his PAX6 gene, and that produces the absence of an iris, a disease called aniridia, so the pupil now nearly fills the eye. Similarly, in the mouse, mutation in one of the two copies of the PAX6 gene partially reduces the size of the eye. Mutations in both copies completely eliminate the eye, the mouse embryo head shown at the upper right. Although Drosophila eyes look very different than mammalian eyes, there's a similar PAX6 gene in fruit flies, and if you make a mutation in that gene, you eliminate the compound eye of the fruit fly. Even more remarkably, if you overexpress this key developmental regulator of eye formation, you can generate new eye tissue in completely different body parts. So this is an experiment that was done by Walter Gehring's lab, taking this PAX6 gene and engineering it to be expressed during leg development in a developing fruit fly. You can see when you overexpress the eye regulator gene, you induce on the leg of the fruit fly a tiny little patch of red compound eye tissue If you do a scanning electron micrograph, that structure has exactly the kind of independent repeated eye unit that you would find in the normal fly eye. And remarkably, you can get that result whether you do the experiment with the fly PAX6 gene or with a PAX6 gene from mice.